Kia ora homa. So it's the first kind of day of the holidays. I normally count the first Monday of the holidays, the first day. I don't count the weekend. So we're going to try the snap idea again. So what I went out and did is I went and bought a brand new thing of yeast. And these are this particular packet is good because they come in lots of little individual packaging. And then the other thing that's good is I went and bought some brand new snap lights as well. So stick around and we're going to go through the whole process again. So if you watched the last video, what I suspected the problem was that it was more to do with, oh yeah, there's a lot of liquid in this one too. These are bigger than the last ones too, so I'm hoping that because I've got a larger quantity of hydrogen peroxide, it's going to help a little bit. So from the last video, I found out that the active ingredient inside a snap light is hydrogen peroxide. And so to do the elephant toothpaste experiment, you need yeast, hydrogen peroxide, and liquid soap and water. So I've got some liquid soap here, and we're going to start off. Now this is dry yeast, instant dry yeast. So what I'm going to do is, normally with this kind of a yeast, it takes a little while to activate. So I'm going to pre-mix this straight away. And what I'll do is I'm going to use the dry yeast mixture as my catalyst. So I'll pour the dry yeast mixture in. So let's start this off. So if you live in the States, you'll probably be pretty familiar with these kind of packets of yeast. They generally come like this in the States, whereas in New Zealand, we tend to get like a little pottle like I used in the last one. Now, my hypothesis as to why it didn't work last time, if you remember, is that my yeast was really old. I would even go as far as saying it had expired. Now, if you're not familiar with yeast, yeast is basically alive. It's kind of like I suppose it's like a fungus or a bacteria. And when you add it to water, that kind of activates the yeast and tells it to come alive again. So just going to do that. Now, yeast likes sugar, and one of the byproducts of yeast and water and sugar is carbon dioxide gas. The yeast eats up all the sugars and releases it as gas, and that's how people make beer and cider and bread and all that sort of stuff. So I've got my mixture there in the beaker. I'm going to put that aside. So now we're going to do the messy hard part, and we're going to separate the hydrogen peroxide. Now, if this doesn't work this time, I would say it's more of an issue to do with the snap lights. So we're trying to eliminate a variable at a time. And my hypothesis, if it doesn't work this time, it's that the concentration of the hydrogen peroxide isn't strong enough to make a reaction. That being said, we should see at least something. Or alternatively, the particular brand that I'm trying to use doesn't have hydrogen peroxide as its catalyst. Now, if you're not familiar with what a catalyst is, just as a reminder, back to little kid science and whatnot, a catalyst is an ingredient in an experiment, a reactive experiment, that makes the whole experiment go off. So the way I teach it to my students is that the catalyst is like the on button for an experiment. So all I'm trying to do right now is saw open that. Don't want that bit, so we'll tip all of that out. We don't need that stuff, because I'm pretty sure that's not the hydrogen peroxide part. And we want the little glass vial that's inside. This bit. Oh, this is fun. It looks like something off The Simpsons. It looks like a radioactive tube of stuff. So this is the bit that I want. Now, the reason why I picked these new glow sticks as well is because they're a lot bigger, that it's easier to get the glass vial out. So the hydrogen peroxide is the bit that's got the dye in it. Now, we might have to try this 2 times. So I'm hoping, from what I've read and researched, this is the bit that has the hydrogen peroxide in it. That being said, it could be the other way around, and the other liquid that I just tipped out could have been the hydrogen peroxide. I don't know for certain, but all I know is I don't want to mix the 2 together, because it'll do that reaction and not the reaction we want, and it'll turn whatever this is or whatever the other liquid is into something else. Does that make sense? So this is the bit we want, and we're going to put it inside our science jar, my famous all-purpose science jar, and we're going to break it inside of here because we want this liquid. Okay? So the next thing we want to do is make up a solution. Yes. So what I'm going to add into here now is I'm going to put this aside so I don't break it by mistake, and I'm going to put a whole bunch of liquid soap in here. Now the reason why we need the liquid soap is the liquid soap is going to capture all of the oxygen and stuff that's being released in the experiment. And what I might do is put just the tiniest bit of water in there, just to help dilute the soap a little bit. Now, I don't expect this to be a massive reaction like my actual elephant's toothpaste video. The only reason I say, see, that's a nice consistency there. The only reason I say that is because I don't 100% think that the concentration of this hydrogen peroxide is going to be strong enough to do that. And also, I haven't added enough ingredients to make that happen. So this is the bit that's a bit sketchy. What I have to do is use some needle-nose pliers. Now, if you're watching at home and you want to try this experiment at home, all these ingredients and stuff I bought from the local supermarket. But because this is a glass vial, you have to get parental help, because you don't want to be like breaking this in your hand and cut your hand and get chemicals all over yourself. So I suggest you do this with an adult at home, show them this experiment, they might get into it, and do it with them at home. And on that note, I'm going to grab my lab coat. Only because I don't want to get my awesome t-shirt destroyed. So thank you, Sato's, for my fantastic t-shirts, one of my favorites. Shout out to Hinata Sato and Haru Sato. So safety glasses, safety first, safety beanie, lab coat, you never know what's going to happen. So I'm going to take these needle nose pliers and break the vial inside of, or drop it, that's entirely plausible as well. I'm going to break it inside here. There we go, broken. Now, obviously the glass is very fragile and flimsy. It's designed that way so it breaks. Okay, now we're up to the fun part. Okay, so it's broken in there. Hydrogen peroxide's mixed with the soap. So now we've just got to transfer all of the stuff outside. And in the comments line, stick whether or not you think it's going to work. Put a comment in the comment line now. See, what do you think? Fail? Yes, no? Let's see what happens. Okay. So I'm going to move for you guys first. I hope your holidays is going all right. Thought this would be a fun thing to do while it's raining. So we've got the deck. No one's around. Well, except Luke. I got Luke Sequera with me. He's my lovely lab assistant today. Oh yeah. Oh, is it?
Yes.
Okay.
So on 0 seconds.
Okay. So remember, we've got our yeast, our brand new live yeast. We've got the hydrogen peroxide and soap and water. So fingers crossed, something's going to happen. Oh, I'm so nervous. I don't know if anything is going to happen. Again, nothing. Okay, one more thing to try. One more thing to try. Now this says to me that the hydrogen peroxide isn't strong enough. That being said, I just want to double check. That the other liquid isn't the hydrogen peroxide part, so I'm going to tip that in. Oh, something is happening. I think it's the other part that's the hydrogen peroxide. So if we start looking now, it's reacted and gone luminescent with the stuff in there, but it's actually fizzing now, or starting to fizz. So it's the other part that's hydrogen peroxide. So what I'm going to quickly, oh man, that's interesting. See it? Interesting. Because it did give a bit of a reaction. So it's got to be the other part that's hydrogen peroxide. Now, what I think the problem is with this part now is that, because it was such a minor reaction, I think that the hydrogen peroxide isn't strong enough. It must only be like 1 to 2%, and we need it to be at least 5 for this to work. So what we'll do is we'll probably come back and try this a third time at a later time, if I can figure out how to get a better reaction. So the cool thing about doing all this kind of stuff is every time you do it, sorry about the light, guys. The cool thing about it is that every time that you do something like this, you kind of figure out the next piece in the puzzle. So it's definitely got hydrogen peroxide in it because there was a little bit of a reaction. Now there's two things. So I already had the other ingredient of the snap light in there, so it probably partly reacted with that and it stopped the reaction that we wanted. 2, the hydrogen peroxide inside the snap light isn't strong enough. So I'm going to have to do some research and see if I can find stronger snap lights or snap lights with stronger quantities of hydrogen peroxide, because in that case it probably will work. So anyway, I hope you found that a little bit interesting, and I hope that answered some of the questions of the people that were watching the video in the last video, because that's going to answer some of the questions that you guys had. So we've figured out that we needed the good yeast. The good yeast worked, I think, and we need a stronger hydrogen peroxide from a better snap light. So if I can get like a really good, like marine quality snap light, like a proper, really good, expensive snap light, it may work. Okay, so stick around and I'll see you in number 3 of this series. Ka kite.